With chapter 6, I'm going to take you on a tour of the cell. We are going to talk about the different type of cells and then how the structure inside the cell fit their function. So this chapter is all about the cell, whether it's be a eukaryotic or prokaryotic cells. It should mostly be a review of what you learned in earlier biology classes. Cells or the cell is an organism's basic unit of structure and function. Many forms of life exist as single-celled organisms, such as the paramecium shown here. Larger and more complex organisms, including plants and animals, are multicellular. In this chapter, we focus mainly on eukaryotic cells, cells with a nucleus. Let's go into cell theory. The cell theory actually dates back to 17th century in 1665, when Robert Hooke discovered the cell by examining very thin slice of barrel cork. Hooke discovered multitude of tiny pores that he named cells. From Latin, cells, cella mean a small room, and this is the image of the barrel cork, very thin slice from the barrel cork. But credit for developing the cell theory is usually given to two scientists, Theodor Schwann and Matthias Schleiden, in 1839. They state that all living organisms are composed of one or more cells. The cells is the most basic unit of life. All cells arise only from pre-existing cells by division. This is a good way to figure out where we are. Just to give you an idea of kind of the size that we are working with. This is an image from your textbook. It shows you different sizes of things, right? So if you remember from chemistry, let's change the slide to this. If you remember from chemistry, we have in atoms. Atoms way down here, it's only 0.1 nanometer. Remember that atoms is indivisible. The meaning of atoms from Latin is the smallest unit of matter that still retains the properties of elements. And then we start to combine atoms to form small molecules. Those small molecules, they covalently bonded with each other to form macromolecules or biological molecules. And we talked about those with chapter 5. Now and up, we have ribosomes. Those are membrane-bonded organelles inside the cells. And ribosomes responsible for making protein. We are going to get that. And then we have viruses. Viruses are a little bit smaller than bacteria. So bacteria is our first prokaryotes that we are going to talk about. So you can see here that from this point down until the end of this arrow, the green arrow, these are things that you can see with your own eyes. And then right here, we have the point at which you can only see stuff with light microscopy. Those things with light microscopy. And here we have electron microscopy. So you can go even further down in size. Note that we don't have on here anything that will show us atoms directly or even small molecules directly. So I don't want you to get back down in that. Chemists use X-ray diffraction or X-ray crystallography. They use scanning tunneling microscopes, special instruments. As a biologist, you student in biology, you will concern only about the light microscope. We have a light microscope. The electron microscope is so expensive, and that's based on scientific research and for medicine too. And we have only light microscope that you work with them in lab. I really just want you to think about these two things that are highlighted in yellow. So in yellow we have bacteria, and the nucleus of the cell. We will talk about the mitochondria. Those are things that we can see with the light microscope. Remember that the nucleus is the only organelle that can be seen with the light microscope. Um, focus on these two highlighted in yellow. The development of instruments that extend the human senses allowed the discovery and early study of cells. Microscopes were invented in 1590 and further refined during the 1600s. Cell walls were first seen, just as I said before, on dead cells of oak bark by Robert Hooke in 1665 and the living cells by Antonio Leeuwenhoek a few years later. So we have two main types of microscopes based on the kind of energy used to view the specimen. We have a light microscope. A light microscope uses visible light is passed through the specimen and then through glass lenses, resulting in enlarged an image seen by the eye when you examine the specimen through the ocular piece. We have three important parameters of microscopy. The magnification. Magnification means that the image appears many times its natural size, or is the ratio of object image size to its real size. Light microscopy can magnify to about 1,000 times the actual sizes of a specimen. Electron microscope can magnify to about 2,000 to 23 or more thousands, 23,000. We have a resolution. Resolution is a measure of clarity of the image. It's the minimum distance two points can be separated and still distinguished, or can still be distinguished as two separated points. You can see here without or magnifying without resolution is nothing. The resolution barrier, when they discovered the electron microscope, at that time they revealed the internal structure of the cells. You can see the organelles inside the cells. Then we have in contrast. Contrast is the difference in brightness between the light and dark area of an image. A method for enhancing contrast includes staining or labeling cells component to stand out visually. Remember that isotopes. When we talked about the isotopes element, those atoms can be labeled any structure of the cell to stand out visually. Just if you remember the yellow area that reveals the cancerous tissue when we talked about the isotopes. And they also used staining, special staining. Here they use hematoxylin and eosine, H&E staining. That stained the nucleus with dark blue or dark purple. And eosine stained three shades, the cytoplasm with the three shades and other component muscles. Here we had neuron stained with the hematoxylin and eosine. You see the nucleus is stained with hematoxylin, dark purple or blue, and the cytoplasm with eosine, it's pink. So they use different staining processing to get the contrast in tissue or in specimen. Until recently, the resolution barrier prevented cell biologists from using standard light microscopy when studying organelles. The membrane enclosed structure, remember that organelles, those are the membrane enclosed structure within eukaryotic cells. So electron microscopy focuses a beam of electron through the specimen or onto its surface. We have two, according to this, we have two types of electron microscopes. We have scanning and transmission. The scanning microscope, electron microscope, is especially useful for detailed study of topography of a specimen. That means the electron beam scans the surface of sample using those specimen usually coated with thin film of gold, and it gives 3D image with scanning. So the scanning is the study of surface of cells or specimen. And then if you see this image here, this micrograph taken with the scanning electron microscope show 3D image of the surface of a specimen. Here, the surface of cell from trachea covered with cilia. Those projection from the cell, we call them cilia. And the transmission electron microscope is used to study the internal structure of cells. The transmission electron aims on an electron beam through a very thin section of the specimen. This micrograph shows section through trachea cells, revealing its internal structure. This image is taken by the transmission electron microscopes. In preparation, the specimen, some cilia were cut along their length, creating longitudinal section, while other cilia were cut straight cross, creating cross section. We can define cytology as a branch of biology concerned with the study, with the structure and function of plant and animal cells. So in general, it's the study of cells. All cells share contain, or they contain those four basic features, or all cells, they share certain basic features. They all bound by selective barrier called plasma membrane or cell membrane. When I'm talking about the plasma membrane, this is the selective barrier that surrounds each cell. Eukaryotic cells, prokaryotic cells, unicellular cells, or multicellular cells. Bring in your mind the biological membrane or the cell membrane. It's a phospholipid bilayer. Bring this image on your mind. Each biological or plasma membrane or cell membrane, it's a double layer, a lipid double layer or phospholipid bilayer with the head, with the hydrophilic head projected outside and inside the cell facing water. And those tails, the hydrophobic tails, they are water-feeling tails. They reassemble themselves in a way that they will hide themselves from water. Okay, so each cell is bonded with plasma membrane, and inside all cells is a semi-fluid jelly-like substances called cytosol, in which subcellular components are suspended. All cells contain chromosomes which carry genes in the form of DNA, and all cells have ribosomes, tiny complexes that make protein according to instruction from the genes. So any form of cell, any cell, eukaryotic cell, prokaryotic cells, multicellular or unicellular, any living cells share those basic features. They all contain plasma membrane, semi-fluid cytosol, they contain chromosome, they contain ribosomes. You might be asking this question: why are cells so small? We're made up of billions of cells, and they are super tiny. And we are not a full singular or giant cell. Why? The problem is the surface area. The surface area is the amount of area exposed on the surface. When talking about cell, the surface area is the size of cell membrane. When we are talking about cell, we are talking about the size of cell membrane. A volume is the amount of three-dimensional space an object occupies. When talking about cell, the volume is how much stuff, cytoplasm, organelles, everything are in the cell. Consider these two cubes, these of different sizes. The smallest cube, 1 centimeter by 1 centimeter by 1 centimeter, that the surface area for cube, if you want to measure the surface area for cube, any cube, they have 6 square, and the surface area for each square is height by width, and the cube any cube has 6 sides, 6 square on each side. So the surface area for this cube, it's height by width by 6. We have 6 square on each side. And the combination, they make the surface area. The combination between those 6 square, they form the surface area for cube. The small cube has surface area 1 by 1 by 6. It will give 6 centimeter squared. Okay. For the large cube, we have 2 centimeter each, so it's 2 by 2 by 6. It will give 24 centimeters squared for the large cube. So the larger cube has large surface area, but also has large volume. The volume is anything inside or the 3D space any object occupies. The volume, it's height by width by length. So for the small cube, we have 1 by 1 by 1. So it's 1 centimeter cubed. For the large cube, it's 2 by 2 by 2, so it will give 8 centimeter cubed. So the ratio, if we will talk about the surface area to volume ratio, the small cube, it's gonna be 6 to 1. The surface area to volume ratio for the large cube is 3 to 1. So the smaller cube has a larger surface area to volume ratio. Let's imagine that these cubes are living cells that need oxygen to survive. For oxygen to spread through the cells, it must diffuse through the surface. As the small cell has more surface area relative to its volume, every part to that volume can be supplied with oxygen at a faster rate compared with the larger cells. It will take too long for the center of these cells or the larger cells to supply it with what it needs. Okay. So cells can't function beyond certain size. As the size of cube increases, the surface area to volume ratio decreases. So do all the math yourselves just to see how things are going when they are increasing in size. Okay. Cells, the basic structural and functional units of every organism. The cells are of 2 distinct types. Okay, we have prokaryotic and eukaryotic cells. The prefix pro mean before, but here it's primitive more than before. So karyon or karyotic mean kernel or the center. That's mean, and here it's mean the nucleus. Karyon mean the nucleus, and pro primitive nucleus. For you mean true or real. Karyon the nucleus. So the eukaryotic cells, they have real nucleus. Here they have primitive nucleus. Organism, and remember that scientists, they classify domains or organisms into three main domain structures system. We have in bacteria, archaea, and eukarya. Remember that bacteria and archaea, they have prokaryotic cells, and eukarya, which include the phylum of plant, animal, fungi, protists, they have eukaryotic cells. And organisms from domains bacteria and archaea, they have prokaryote, and eukarya, they have eukaryote. Protists is an informal term referring to a diverse group of mostly unicellular eukaryotic cells. We will start with prokaryotic cells. Those are characterized by having no nucleus. Just as I said before, they have primitive nucleus. Their nucleus is only the form of circular DNA that's concentrated in a region that is not membrane enclosed, called nucleoid. So the DNA is simple circular DNA molecule concentrated in a region called nucleoid. And they don't have membrane bonded organelles. They have cytoplasm or cytosol that are bonded with the plasma membrane. Just as I said before, the four main features of all cells, they have chromosomes, or they have genetic information, they have cytoplasm, they have plasma membrane, and they have ribosomes. So bacteria have only ribosomes. Only organisms of domain, as I said before, to bacteria and archaea, they exist as eukaryotic cells. We have eukaryotic cells. Eukaryotic cells, they characterized by having DNA. That DNA in the form of chromosomes bonded with nuclear envelope, so they have real nucleus in the center of the cell, and they have membrane-bonded organelles. The cytoplasm occupies the region between the nucleus and the plasma membrane. Eukaryotic cells are generally much larger than prokaryotic cells. And remember that eukarya, which include the protists, fungi, animals, and plants, they all have eukaryotic cells. So I want you to memorize the difference between prokaryotes and eukaryotes, just like your name. For eukaryotic cells, in addition to the plasma membrane at its outer surface, eukaryotic cells have extensive elaborating arranged internal membrane that divide the cell into compartments. Those are the organelles. So they have an organelles, membrane-bonded organelles. They have a plasma or a biological membrane, just as I said before, when we are talking about the membrane, we are talking about biological membrane that is composed of double layer of phospholipid bilayer. So this is the membrane. It's composed of double layer of phospholipid molecules arranged in this way. The basic fabric of most biological membrane is the double layer of phospholipids. They are embedded within. They are phospholipid embedded within the membrane and attached to its outer surface, proteins or enzymes. They contain enzymes just like the membrane of mitochondria. We have two types of main two types of cells of eukaryotic cells that we will talking about. We will talk about plant and animal cells and the differences between them. Consider that the plant cells have cell wall. Cell wall, you remember from the macromolecules, we talked about the carbohydrates and the structural material, building material cellulose. So the cell wall is the outer layer that maintains the cell shape and protects cells from mechanical damage, made of cellulose, other polysaccharides and proteins. Plant cells, they have cell wall in addition to the plasma membrane they have in cell wall. Plant cells, they have in plasmodesmata. Those are cytoplasmic channels through the cell wall that connect the cytoplasm of adjacent cells. Plant cells, they have chloroplasts. Chloroplasts, the photosynthetic organelles, convert energy of sunlight to chemical energy stored in sugar molecules. Then we have in animal cells. Animal cells, they contain, or let's back to the plant cell, they have a central vacuole. Remember that the plant cell, they have central vacuole. This is a prominent organelle in older plant cells. The function includes storage, breakdown of waste products, and hydrolysis of macromolecules. Enlargement of the vacuole is major mechanism of plant growth. Then let's go to animal cells. Animal cells, they contain lysosome. Lysosome, this is in membrane-bonded organelle. Contain or has digestive, those are digestive organelles with the digestive enzyme. They digest the macromolecules, and we have centrosome. Centrosome, region where the cell's microtubules are initiated, contain a pair of centriole, and we have in flagellum. This is the singular flagella, or flagellum the singular, and motility structure. This is our motility structure present in some animal cells, composed of cluster of microtubules within the extension of the plasma membrane. Okay, we will study the organelles or put them in 4 functional categories. We will start with the first one. We'll start with the genetic control of cell. We have 2 organelle. Nucleus is which houses most of the cell's DNA, and we have a ribosome, which use information from the DNA to make proteins. So on the first stop of our detailed tour of eukaryotic cells, we will talk about the nucleus. The nucleus, as I said, it contains most of the cell's genes and is usually the most conspicuous organelle. And the nuclear envelope, it's surrounded by double membrane. The nucleus is surrounded by double membrane, and we call this double membrane nuclear envelope. And the nuclear envelope is composed of double membrane. Double membrane, it's continuous with each other, and each of those membrane consists of lipid bilayer. So the nuclear envelope is double membrane. The double membrane, each a lipid bilayer. This is the nuclear envelope. This is the nucleus, and this is the nuclear envelope, and this is the endoplasmic reticulum attached to the nuclear envelope. And this is double membrane. Each of this layer is lipid bilayer with associated proteins and are separated by space. This is the space between the double layer of membrane, cytoplasmic membrane. The envelope is perforated by pore structure. This is the pore structure those. The inner and outer membrane of the nuclear envelope are continuous, as I say, and an intricate protein structure called pore complex lines each pore and plays an important role in the cell in the cell by regulating the entry and exit of proteins and DNA and RNA between the nucleus and the cytoplasm. The nuclear site of the nuclear envelope is lined by a nuclear lamina. This is the nuclear lamina. It's beneath the nuclear envelope. This nuclear lamina maintains the shape of nucleus by mechanically supporting the nuclear envelope, and it's made of a network of intermediate filaments or protein filaments. Within the nucleus, the DNA is organized into discrete units called chromosomes. Each chromosome contains, this is the chromosome, each chromosome contains a long molecule of DNA associated with many proteins, the histone protein, those the histone proteins. The complex of DNA, this DNA molecules, and the protein together, we call them chromatin, the building material for chromosomes. When the cell is not dividing, stained chromatin appears as diffuse mass, and the chromosome cannot be distinguished from one another. Only during division, the chromosome condensed or the chromatin condense and coil and becoming thick enough to distinguish a separate region as chromosomes. The primary function, this is the chromatin, okay. The primary function of chromatin is packaging long DNA molecule into more compact, denser structure. This prevents the strand from becoming tangled, and also plays an important role in reinforcing the DNA during cell division. This prevents DNA damage and regulating gene expression and DNA replication. Inside each nucleus, there is prominent structure within the nucleus, or we can see it in non-dividing nucleus, is a nucleolus. We have a nucleolus inside each nucleus, which appear through the electron microscope as a mass of densely stained granules. Here, a type of RNA called rRNA, ribosomal RNA, is synthesized from genes in the DNA. Okay, so remember that the nucleus is composed of nuclear envelope with pore system. We have a nucleolus. We have the gene, the genetic information in the form of DNA or chromatin. DNA with the proteins form the chromatins, and the chromatins form chromosome during division. Only during division we can see the chromosome in this form. Okay, inside the cell we can see a mass of a diffuse mass of chromatin fibers only, and the chromatin fiber is composed of DNA molecule with histone protein together form those small beads of nucleosomes, and then this nucleosome packaged into or condensed and coiled to form the chromosomes. So we can see the chromosomes only during division. In the normal non-dividing cells, we see only the chromatin fiber. Remember that. The second organelle in genetic control of the cell, we have a ribosome. Ribosomes are complexes made of ribosomal RNA and protein. They form from ribosomal RNA and protein, and the ribosome carry out protein synthesis in two locations. We have free ribosomes that are suspended in the cytosol. They make most of the protein that we use inside the cell. And we have bonded ribosomes, usually associated with the endoplasmic reticulum and the nuclear envelope. Those make proteins for export or for cell organelles membrane. Ribosome play important role during protein synthesis. If you see here, this is the gene expression, how the RNA would formed inside the nucleus, make a copy from genetic information from the DNA molecules, and then this messenger RNA have a message from the DNA to the protein factories inside the cytoplasm to build the required protein needed by the cell. Okay. After RNA or messenger RNA have been synthesized inside the nucleus, they passed through the pore complex I mentioned before, that's found in the nuclear envelope. They pass through this pore complex into the cytoplasm. Inside the cytoplasm, or attached to the endoplasmic reticulum, we have a ribosomes. Ribosomes composed of two subunits of special proteins. One of them is SB50 and the other is 30S. Those combine with each other, and then they build new protein or polypeptide. The amino acid sequence on each polypeptide that been synthesized, it's translated from the sequence that the messenger RNA brings from the DNA. So they have sequence of amino acid depends on the sequence of nucleotide of DNA molecules.